Am I audible? I hope. Am I audible? Okay, fine. Yeah. Thank you, everyone. I'm Pratik Dutta. I'm a senior research scientist at Stony Brook University. I'm at Department of Biomedical Informatics. So, coming up with background of computer science, I'm still learning of biology, and I'm trying to maintain the gap or complete the gap. How we can address as a computer scientist to gap those biological things. So today my talk is how a language model can help or understand the impact of the variants. So it all starts about how a genomic language model can understand the context of the human genomes, and once it understands the context of the human genomes, how can it do different downstream tasks. So one of the downstream tasks where I am mostly focusing on right now, how a language model understand the variants effects. So here my entire task or entire focus into the non-coding region, which is the regulatory region. So when we actually check the variants association or position there, so we saw like most of the variants is not not like randomly associated into the non-coding regions, but if you can see the figure, it is actually more concentrated near to the transcription start sites. So from starting from there, we actually saw like, okay, while those are variants are actually present into near to the TSS region, how those variants is actually affecting different regulatory regions. So based on this motivation, we started our building one framework, which is totally based on one genome foundation language model, which is called DNABird. That DNABird is actually developed in our lab. So we built this framework totally based on the DNABird. We named as a DIBV regular. So this is the first we'll talk about the what is the computational approach of this DIBV regulum. So in the DIBV regulum pipeline, we have the total three architecture. Like if you divide the total pipeline into three parts, so there is total three parts. So one is the fine-tuning stage. Second stage is the visualization module, and third stage is the prediction module. I'll come for each module one by one. So as I already told, like it's entered the total pipeline is based on the DNABird module, and in this pipeline we made around 702 fine-tuned models. So each models is for a specific transcription factor binding sites or histone markers or splice sites. So I'll come one by one. And so as the entire pipeline is based on the DNABird, which is the one of the genome foundation model. So here is one slide, which is just a brief overview how the language model, DNABird language model works. So DNABird actually divide into two stages. One is the pre-training, another is the fine-tuning. So if I just give you the explanation, what is the pre-training and fine-tuning? So pre-training is it's understand the context of the biological language, and fine-tuning is kind of very precise things. Like if I think about a language model, it actually captures the small like local context and the global context. So global context is coming from the pre-training region, and the local context is coming from the fine-tuning strategy. And now this is a brief overview of the DNABird, and now we are coming to the fine-tuning stage. So as we are actually predicting the variant effects into different regulatory regions. So here in this pipeline till now, we actually completed for the 700 transcription factor binding sites and 2 splice sites, like one acceptor and the donor regions. And for each fine-tuned region, how I did the fine-tuning, I have the figure in the later picture. But for each data sets, we are creating one positive set and the negative set, and based on that sets, we actually created the fine-tuned model. So this is a schematic diagram of the fine-tuning model. So it's a one. It is a diagram for a one particular model. So it is all the models or all the models follow this flowchart. So let's suppose how we build it. Let's suppose we are talking about the acceptor model, or let's suppose we are talking about the CTCF transcription factor binding sites. So in that case, first we create the positive set and the negative set for the fine-tuning model. once we create that thing, we do the tokenization. Here we did the 6-month tokenization, as we already checked for different token, tokenization, like 3-month to 6-month into the DNA bot, and we saw for the 6-month it is showing kind of better results. So we started with the 6-month. And once we got the tokenization, we took the pre-trained DNA bud model, and we just changed the with gradually changing the attention layers, or like entire hidden layers weights, and finally we got the fine-tuned model. So for this total 700 models, we actually run over the A100 GPUs, and it will take more than 7 months time to get all those 700 models. And among those fine-tuned models, so there is another part which is more important also, that is the attention score or visualization. Like when we are talking about the fine-tuning, it's also find out which are the motives, which are the important motives are there, which best the model is actually performs well. So this is the first thing. Is the next thing is the downstream task. Like once we get all those 700 models, next comes our downstream task. So in here, our downstream task is the variant predicting variant effects into the regulated regions. So for that, here till now, right now, we have only applied for the GBM cancer patient. We got 190 GBM cancer patients from TCGA GDC portal, and we are actually applying this framework for other cancer patients from the GDC cancer portal. So we completed the GBM cancer patient. We are working on the breast cancer, and we'll apply for other cancer also, plant cancer things also. Now, we started with that 190 cancer patients, and we focused only the somatic mutations. Among that somatic mutations, we took all the substitution and indels. And this is the one schematic diagram of the how we did the prediction task. Like, let's suppose we have already the fine-tuned model. We have around 700 models. Now, when we trying to find out if that variant is predicting the region or not, we are actually passing a pair of the sequence. One is wild type, another is the mutated sequence. And based on our DNA bud score or probability, we actually calculate two quantitative measurement. One is score change, another is the log odd ratio. Based on that, we find out which variant actually affecting the regions or not. So that's the computational part. Like, we based on DNA bud, we find out the which variants is predicting the regions or not. And once we got all those candidate variants, we did different biological validation. Though we did not do any kind of experimental validation, we are still looking for some collaboration so that we can do the wet lab experiments. But we still, whatever we did, yeah. So I just saw the two schematic diagram of the fine-tune and downstream task. So this is the overview. So I am not going into the first part, but if you see the downstream task, so right now we did total 4 downstream tasks till now. So first thing is the creating the candidate variants, whatever we got from the DNA models. So candidate models, we make a database. Second thing is, once we get the candidate variants, we check how those candidate variants actually changing or associate with the attention regions. Third is motive finding, like how those models actually find different known motifs into the transcription factor binding sites. And finally, survival analysis. So all those kind of mostly biological analysis and validation is done based on the publicly available data sets. So before going into the publicly biological validation, so this is the performance of our models based on the computational aspect. So among that 700 models from the transcriptive-based models, we did not consider all those models. We consider those models which having highest accuracy, like high accuracy. So our threshold is right now 85%. So when we actually put the threshold of 85%, we got around 460 models, TF models, and 2 splice sites. So the first figure is Now, among the 460 TF models, only 4 we got from the histone markers model and all the others from the TF. So the green one is showing for the TF models and the red one is the for the histone markers. So first figure is all the 460 models, accuracy, precision, recall, other performance metrics. Second and third figure is showing the UC curve for the top 15 models for the TFs and the only 4 Wheatstone markers we found from the 460 models. So that is the that one. The last 3 is like the first 2 is the top 15 TF models for the accuracy things, and the last one is the for the splice sites. Now, coming into the other part, like when I'm talking about the fine-tuning part, so the fine-tuning contents 2 stage, right? So one is the creating the fine-tune model, another is the visualization model. So the last slide is for the fine-tune model's performance. This part is the when we actually check or tried out, okay, if that models find out any known motifs of those TFs regions. So we checked there are several known motifs is actually matched with those predicted motifs. So we used one attention visualization module, and from there we actually checked or compared with the known motifs databases. So we compared with the JSPER 2024. So if we just so I'm just giving here only 6 examples, but we have a lots of examples. Yeah, so if you see the top one is we actually get it from the Jasper motifs, and the bottom one is always getting from the our DB regulum predicted motifs. So we found we found a good match with the less p-value. This is the how many candidate variants we found based on different splice sites model and TF-based models. Once we get the candidate variants, we actually checked or associated with the known databases. So we also associated the DB-SNP databases here. So here, this plot is only for the SNPs, like for the substitution variants. So where you can see the no model actually find most candidate variants which actually associated most of the DB-SNP entries. This is the same for the indels variants. This is the 2 figures where rather than finding the here we are actually mostly focusing on the models where you can see the CTCFL which having the accuracy around 98%, but it's getting only 4 candidate variants and it is associated with the 3 DB-SNP association. So now I'm coming into the little more biological validation things when we get all those candidate variants and we find out all the biological, all the foundation models and everything. Now it comes about whenever we got the candidate variants, what is the position of those candidate variants? If that candidate variants located in a known motif region or not. So we got one, I mean, we got a lots of example. Here I'm showing one particular example here. And so here the example is from the ETS1, where you can see, yeah, can you see my, what is the reason? Yeah. And so this is the so for the when we create the foundation model, so we actually consider around 300 base pair of sequence, but we are plotting here only 20 base pair regions so that we understand. So we can see here the reference nucleotide is actually changed to C. So the mutation is like G is actually changing to C, and our probability based on the DB regulum, it's the probability changes from 0.99 to 0.31. So like only changing only one mutation or probably incorporating with one mutation, our model or our framework says, okay, there is some changes into the pattern or some composition of the hex marks or 6 marks, so that the model says, okay, your probability is actually changing from 0.99 to 0.31. So when we checked the position of the mutation, we actually see that mutation, like that variant is actually present into the known motif region. And when we also just check with the UCC genome browser, we see that that variant is also present into the near to the some known unknown protein. I don't know if you can see that's the GATA D2B. So GATA D2B is also responsible for different brain associated works or brain associated functions. So it could be like from here we can just say, okay, this variant is present into some motif region which either inhibit or prohibit the binding sites or binding properties, and which also changes the probability of those reference probabilities to the like reference probabilities from the alternative probabilities. So that is we found out this type of some candidate variants which are actually found, located nearby some known genes which are actually associated or responsible for different brain associated biological functions. That could be some cancer, that could be some rare disease like Parkinson's or other things. So next thing we did different survival plots based on those based on those candidate variants. So what we did, we actually divided the, we have 190 patients, so we actually divide those 190 patients based on the variant present or not present. So we saw two types, both types of tendency. Some cases we saw the variant present into the group which is showing good survival. Sometimes we saw the variant present into that group which is showing in very bad survival. So we actually checked, one of our lab member, Matthew, he's a MD PhD student, he's a clinical person. He actually checked like if that variant is present any known gene region or not, and how the other things. So when he checked, so he found that most of the we got like a good amount of candidate variants which is actually present not over the any genes, but very I should say proximity of the genes which could either inhibit or prohibit the different cancers, different cancer genes or associated cancer genes. So this is another, we check for the all those TF-based models. We found few survival like hazard ratio, some suppose hazard ratio is less, some suppose some hazard ratio is more than one. Yes, I don't know if I can see play that video or not. Is it running? Yeah. So though we created the candidate variants, but we created one database also, as we said. Oh, fine. Yeah. So we created the databases also, but we did not publish it, but we already made it because the work is still into the, we are writing the manuscript, we are into the preparing the manuscript and submitting the manuscript. But the database I already completed, where we can show what are the candidate variants we found, how those candidate variants is associated with the known genes or not, how those survival plots are actually showing. So those are the few things where we can show it into the candidate, into the databases. Yeah, hopefully when we submit the or publish the paper, we also publish or make available these databases. Finally, I would say the key takeaways, like as a computer scientist, when I create those models, so what the first question when we got, when we presented some some forum where it's a kind of mixture of biologist and the computer scientist, the biologist said, what we can get from this model. Okay, your model works good, but what is the biological interpretation we can get? So we made the model or this framework in a way so that it could be generalized, or we can apply that model of any kind of cancer or any kind of rare disease patients. So what we need only the VCF file where we can have the mutation information, and from there we can find out if that variant is affecting or changing any functionalities to the different known regulatory regions. That could be splice sites, that could be promoters, that could be other TFs regions. So we're trying to make that thing as much as generalized it's possible. We did not compare, we are still comparing with the known models, which is the DeepSea and BasenG2, but as I don't have the result, I did not put those results right now. Yeah, so this is my amazing team members. Without them, it is not possible. Though I am a computer scientist, this lab has a good mixture of the biologists, statisticians. The Ramana WD is the lab PI, and who actually made his brainchild is the DNABot and DNABot2, where based on those things, this framework is actually made. And thank you so much, everyone. I'm happy to, if you have any questions, I'm happy to give you the answers. Yeah.
Probably answered these questions, so sorry, bear with me. You were predicting the effect on the coding region.
Non-coding.
No, the effect of the non-coding on the coding. Like you were predicting full change of gene expression.
Yes.
That was the
yeah. So suppose a variant is present in splice sites. So if that variant presenting into the splice site is changing the splicing effect or not. So I am actually checking that thing, or probably if that variant is present into the any TFBS region. So if that variant is actually affecting the binding functionalities or not. So I am actually predicting those things. So first,
it's basically like an EQTL or an SQTL.
Yeah, so we did not do the EQTL, but we'll do the EQTL. So right now we only check if that variant is having any association with the CleanVar or DBSNP. But for the more biological analysis, we are still doing those things. So we have, we did not do any kind of experimental validation till now, but they are Matthew and Rekha is also here. So they are actually doing all kind of this biological validation things.
So I'm not sure if you addressed this, that was just for clarification,
Yeah.
Thank you. I'm not sure if you addressed this or not, but I'm curious, if you train on one cancer, can you predict another cancer?
Yeah, obviously we can do it. I mean, but the thing is, so right now we only did it for the GBM cancer patient. So what we are doing, we took it from TCJ. So we are looking for some other cohort of the GBM cancer patient, and we are trying to check how this model is actually performing.
But why not test on other cancers?
We can do it, but our idea is we should. I mean, the model is generalized. We can obviously do it, but we'll try to do it if it is a GBM cancer patient. So how those models actually shows the performance of the other cohort from the GBM, and also it is kind of GBM to GBM, and also we can check GBM to breast, lung, and other things also. So starting with from GBM to GBM, and probably after that we can go with other pan-cancer things. Thank you. Yeah.
Nice. So you have 700 or so fine-tuned TF models, and you said 460 or so are working fine, right? Did you look at whether those are the transcription factors that have a well-defined motif, and the ones where your model doesn't work well below that threshold are things where you really don't have any motif in these motif databases? Like
So-
what separates those two?
Yeah, so actually how we separated, we first created all those 700 models, but we did not check for the models which have the accuracy less than 85% or which is a very less F1 score. So suppose in that probably we have around 170 or something. So what we are doing, we still did not check the known motif finds of those very bad performance models. But what we are doing right now, we first check why the model does not work good. So it shows if we started about 700 models, among that 33 are histone markers were there. And among that 33, only 4 histone markers we got greater than 85%. Now, when we actually check the data, it says we saw the data is very noisy most of the cases. And when we see the chunks what we took around 300 base pairs. So for 173 models, what we are doing two things. One is we are increasing the sequence length from 300 to 500 base pairs to see if it could capture more context. Second thing is rather than only using the genome data, genomics data, we are also adding some physiochemical properties of the sequence to see if the model can understand more, like from those data, from those information, it could model and learns better than only the genomics things. And I think the other part, what we'll do as the TF-based models, when we are saying we are creating the data, how we create from the peak, we are taking the like right now 300 base pairs, so 150 base pairs positive, 150 base pairs negative. So it could be possible that the peak value is not that strong, so that, but we took it as a training data set. So we are actually checking some peak strength, like checking if possible, if we get some information of the peak strength, so that we can create good data. Because all the what we believe, or at least I personally believe, whatever we make the model, all depends upon the data. So if the data is good, the model will work. Yeah. So yeah, thank you. Yeah.